Welcome to Online Lecture 5, which covers Chapter 9. In the last chapter, Chapter 7, we started covering mitosis, which involves the division of the nucleus during cellular reproduction. We sort of skipped over a phase called the S phase as part of the cell cycle that precedes mitosis, having referred to that S phase as the DNA synthesis phase. The lecture today is going to cover what happens during that synthesis phase, that is DNA replication. It also covers the topic of telomeres and DNA repair. Those topics I just mentioned are all covered in section 9.2 of your textbook, which means that as you do your reading and studying, this is the section that you need to focus on. So before we start studying DNA replication in detail, I thought we would begin with some history about how we learned the function of DNA. Before the structure was known, biologists had noticed something they called the transformation factor. And this was first reported when studying two strains of bacteria called pneumococcus. The first one was the S strain, and the S strain caused fatal pneumonia in mice. The pictures that you're looking at in these slides all show the smooth strain, the S strain of the bacterium. And if you look at these cells, particularly up in the right-hand corner of your screen here, you'll notice the bacterial cells inside of them are surrounded by a big sheath. The cells, the other strain is called the R strain, and those cells actually lack this sheath that you see here. The sheath is also visible by this sort of rough edge around the outside and the bottom of your screen, that cell that's in the bottom of your screen. And then this is what the bacteria actually look like growing on the plate. So that smooth outer covering might not look like much, but it was actually pretty fatal to the mice. You can think about it this way. I don't know if any of you have ever played a game called Greased Watermelon. Sometimes people play it at camp. But during the game, what happens is a watermelon is greased and then gets thrown into the swimming pool, and children have to dive in and try to grab it. Well, trying to grab a greased watermelon is not very easy, and similarly, trying to grab a bacterium with a smooth outer shell is very difficult for the mice's immune system, and that's why these S strain bacteria caused a fatal disease, whereas the other strain, the R strain, which lacked that coating, did not. What we know now is there's a genetic basis for this, and that's that there's a single base pair change in the sequence for the cell wall gene that can convert the R strain into the S strain. But this was not known back then. And instead, what happened is that there was a series of experiments that showed that there was some kind of transformation factor, that this S strain could pass to the R strain. Even when it was dead, even when the S strain was dead, it could pass this transforming factor to the R strain and cause it to actually become an S strain and cause fatal disease in the mice. Now, the microbiologists that were studying pneumococcus were very curious about this transformation factor and really wanted to know on a biochemical level what substance it was that could literally turn one strain of bacteria into another. And so they set up a series of experiments to try to determine just that. And these experiments are diagrammed on this slide. It's a very famous set of experiments known as the Avery-MacLeod-McCarty experiment. And basically what these scientists did was take an extract from heat-killed S strain bacteria. So these are the bacteria that caused the fatal infection, the fatal pneumonia in the mice. And they killed those bacteria, and then they broke open or lysed the cells. And they did a series of experiments where they systematically destroyed the biochemical components of that extract to try to figure out which one of those components was responsible for actually transforming the R strain into the S strain during their experiments. So let's take a look at what happened. And in each of these experiments, what the scientists did was to take this extract from the S strain of bacteria. They treated it in a variety of ways, and the treatments are shown here as number 1 through 5 on the top of your screen. And then they added that treated extract to the type R strain. So this was the strain that did not cause pneumonia. And then they took that mixture and they injected it into the mouse. I'm not going to go over each one of these experiments in detail because it would take too long and it's really outside the purview of our class. What I want to point out to you, though, is that when they destroyed the lipids and the polysaccharides in the extract from the S cell, combined that to the R strain, then injected that combination into the mouse still died. And it was only in their very last experiment, which is shown here on number 5, where they actually took an enzyme called DNase, which destroys DNA, and treated the extract from the S bacteria with it, and mixed that with the R strain bacteria and injected that in a mouse. It was only in that experiment where the mouse actually lived and there were no S strain bacteria recovered from that living mouse. And so what they concluded from this experiment was that it was DNA that actually was the transforming factor, and therefore it carried hereditary information. We now use the term transformation experiment as one in which DNA from one strain of bacteria genetically transforms another strain. Avery and MacLeod and McCarty were the first ones to observe this phenomenon, but it's actually used extensively in the lab now. These pictures I'm showing you on this slide are kind of a little bit disturbing and maybe not the most ethical experiments, but they do demonstrate that you can take DNA and pass it through any cell membrane, including egg cells, and passing this DNA through a cell membrane can genetically transform an organism, resulting in what we call a transgenic form of that organism. So, for example, people have put in genes for an ear in mice to grow ears in weird places, and there are a variety of experiments where a protein called green fluorescent protein has been placed into mammalian tissues, and those mammals that resulted actually glowed bright green. It's kind of interesting to look at, but I certainly wouldn't want to volunteer for any of those experiments. Either way, this is what we call the transforming principle, and all of this supports the idea that it is DNA that carries the hereditary material. So you should recall all of the facts that are shown on this slide as material that we covered in Chapter 3, which is where we discussed the structure of DNA. We didn't mention the fact that the structure of DNA was first proposed by James Watson, who is shown here on this slide, and Francis Crick, who is shown over here. These were two scientists that were working in Great Britain at the time, and we know now that they proposed a fairly simple but elegant structure. And the reason we just want to revisit it and think about it now is that there are some key features of the structure of DNA which gave some hints in terms of the way in which this molecule might be replicated. And this fact was also noticed by Watson and Crick. So here's their famous paper that was published in 1953 in the April 25th version of the journal Nature, which is a very prestigious journal. And I just want to call your attention to a sentence here at the very end of their article, and they say this: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." The particular part of the structure that Watson and Crick referred to as suggesting the copying mechanism for DNA is the complementary base pairing between the strands. We're going to turn our attention now to some other features of the structure of DNA that will help us understand some material in later chapters, but also in this chapter, the process of DNA replication. So we're going to look at 6 key features, some of which are review, so I won't go over in detail, and some of which are new. The first one, which is review, is that the nucleotide bases are on the interior of the 2 strands and the sugar phosphate backbone is on the outside. From a chemical perspective, this makes the backbone have a very different chemical characteristic than the interior of DNA. For example, the sugar phosphate backbone has an overall slightly negative charge, whereas the interior of the strand where the base pairing occurs is actually slightly hydrophobic. The second feature is that of complementary base pairing, and this is where we have each base that pairs specifically with either a purine or a pyrimidine in a very specific fashion. We also know that DNA is double-stranded and that it has a uniform diameter. So even though it's twisted, if you were to untwist it and look at it, you would see that there was the same distance between the sugar phosphate backbone at any point along a double-stranded piece of DNA. I mentioned before that this double helix structure of DNA is actually the same one that would result if you took a ladder and twisted it. Well, if you think about it, you can twist a ladder in either one of two directions. And we use the term right or left-handed to describe the direction in which a twist can actually occur. The twisting that occurs in DNA is in what's called the right-handed direction. And we call the twisting motion of DNA a right-handed twist, or it's a right-handed double helix. Because if you were to take your hand, like so, with your thumb pointing up, and wrap it around the DNA double helix, you'd find that if you do it with your right hand, that your fingers run the same direction as the grooves. Whereas if you were to take it with your left hand to do the same thing, your fingers would run against the groove. And so for that reason, we call DNA a right-handed double helix. We also learned in Chapter 3 that the two strands of the DNA double helix are oriented in what we call an anti-parallel fashion, which means that one of them is upside down relative to the other. It also means that any particular strand of DNA actually has a directionality to it. The directionality of each strand of DNA actually arises from the fact that each nucleotide has a directionality associated with it. So I'm going to try to give you a simple example that I hope will help you understand what we mean when we talk about DNA having direction, and also we talk about DNA having anti-parallel strands. So let's pretend for just a moment that my hands are nucleotides. And this part of my hand are going to be the hydroxyl groups, and my thumbs are going to be the phosphate groups. So you can see here there's a phosphate end and a hydroxyl end. And for our terminology, the phosphate end is always called the 5' end. It has to do with the number, the carbon number that the phosphate group is attached to. That's why it's the 5'. And we also see the hydroxyl end as referred to as the 3' end. So here's the 5' end, and here's the 3' end. And you can see that I could arrange those nucleotides in any way I want, but actually they're arranged in a very specific way, so that during DNA synthesis what happens is that you have an existing nucleotide and another nucleotide comes in and is added to it in this fashion, and only in this fashion. You might think that the opposite reaction, where you have a phosphate group with a hydroxyl being added to it, is the same reaction. It's not. The DNA polymerases that catalyze this reaction can only catalyze the reaction of the phosphate group being added to the hydroxyl group. So every strand of DNA then, pretend this is my polynucleotide chain here, has directionality associated with it. Here is a phosphate group. Which end is that? If you said 5', you're right. Here's the hydroxyl end. If you said 3', you're right about that. So one of our chains is going to be oriented in this fashion with the 5' end sticking out. The other is going to be oriented in this fashion with the 3' end or the hydroxyl end sticking out. And that's what we mean when we say that the DNA strands are organized in an anti-parallel fashion. I want you to keep this idea of 5' to 3' synthesis in mind as we go through this chapter and the topic of DNA replication. The last idea of DNA structure that we want to talk about today is actually a new one that I haven't introduced before. And this is the idea of areas on the DNA double helix that are referred to as the major and minor grooves. The grooves actually just refer to the space in between the sugar phosphate backbone that results from the twisting of DNA. And you might have been able to guess this, but the major groove actually refers to the space that is the largest, and the minor groove refers to the space that's a little bit smaller. And if you look closely at this picture, you can see that the way that the twisting occurs results in a bigger major groove and a smaller minor groove. The differently sized spacing of the grooves results from the fact that the spaces between the twists of the helix are not even. You can see that very well from the animation here on the bottom of your screen. And so coming through right now on the left-hand side of your screen, there's a minor groove, and as it twists around, if you keep watching it, here comes the major groove, big space right here. These grooves are actually very important because they govern the chemical interactions in which DNA is able to participate. And the way that these grooves actually govern the interaction that can take place is a function of the actual sequence of bases that is exposed in a particular major or minor groove. In one of these grooves, the outer edges of the base pairs are actually accessible for hydrogen bonding. And if you look at this animation, you'll see as the DNA spins around that, particularly in the major groove, those bases are actually fairly accessible. And of course, we know that there are two different kinds of base pairs. There are adenine-thymine base pairs and guanine-cytosine base pairs, and they're chemically different. Because of the difference in the chemical characteristics of an AT bond and a guanine-cytosine bond, and the fact that DNA sequence varies along the length of its molecule, the actual chemical signature inside each major and minor groove is actually different. And what I mean by chemical signature, I mean the orientation and number of hydrogen bonds in which that groove is available, in which the bases in that groove are available to participate. So one of the things that we're going to see as we go on this semester, that many of the processes that we're going to cover, like DNA replication in this chapter or gene expression later on, involve DNA-protein interactions. So a protein binds to a DNA and either stops a process or initiates it. Well, that binding usually occurs in the major groove of DNA, and a particular protein usually can only bind in a specific major or minor groove. And this specificity of binding is governed by the particular sequence of bases that are exposed in the major groove and the specific sequence of amino acids on a protein. So those two things have to line up in order to allow hydrogen bonding to occur between them so that the reaction can take place. And this is a very specific kind of interaction that keeps proteins from binding anywhere. And this is very key to having the appropriate DNA-protein interactions that are necessary for cellular processes. So in addition to exposing this very specific chemical signature of a groove, the grooves are also important because they just form a physical space where proteins can actually interact with DNA. And this slide is showing three different kinds of DNA-binding proteins. And if you look at this carefully, you'll see that in every case where the protein is interacting with the DNA, it's doing so in the major groove of that DNA. And as I just explained on the previous slide, this kind of interaction requires that there be a very specific sequence of amino acids present on the protein and a very specific sequence of nucleotides on the DNA, those nitrogenous bases that are actually exposed to allow for hydrogen bonding. Finally, in a familiar concept that we've discussed several times this semester, we see how the structure of DNA gives rise to its function. And DNA has four primary functions, all of which arise from its double helical structure. The first one is that DNA stores the genetic information. It consists of very long strands with millions of nucleotides, and we know that the sequence of bases encodes huge amounts of information, and that the information is not stored in the sugar phosphate backbone. We'll see in part 2 of online lecture 5 how the complementary base pairing of DNA gives rise to a very precise replication process that precedes cell division. We'll also see a little bit later on this semester that the structure of DNA, relying on this specific sequence of bases, actually does make it susceptible to mutations. And so a simple change in information, for example, a simple alteration in a sequence, can actually have some fairly serious genetic consequences. And then a little later on this semester, we'll actually see how it is that the structure of DNA, or the genotype, gives rise to what we actually see, which we call the phenotype. This concludes online lecture 5. Be sure to also watch parts 2 and 3.